You all for coming. We really appreciate your time and we really are really excited to have our April Our Ladies Melbourne event. I would first like to begin by acknowledging the traditional owners of the lands on which we are meeting today. For me, that is the Wurundjeri people of the Kulin Nation, and I pay my respects to their elders past and present. I also acknowledge the traditional custodians in the lands of which you are all joining from today and extend my respects to their elders past and present, as well as to any First Nations people with us. So, who are we? If you don't know by this point, I will let you know. So, Our Ladies Melbourne is a registered nonprofit organisation and a local chapter of Our Ladies Global. Our aim is to promote gender diversity in the art community, both in Australia and worldwide. We have a website that houses all of our main goals, as well as updates on our events, our committee, our legacy, and some resources that we found really interesting. We also have an expression of interest form here in the navigation bar in the About Us tab. So if you have a seminar topic, a workshop, or small talk in mind that you'd like to give at an Our Ladies Melbourne meetup, please fill out that form and we'll get back to you as soon as we can. You can find us on various social media platforms, and we have our own Slack community where we hope that we can foster an engaged environment for our tips and tricks, promoting our events and others of interest, and so on. I'd like to introduce and welcome our 2025 committee. We have Harriet, we have Megan, Kathleen, Dai, Kirsty, Felicia, Serish, Mansi, Lauren, Cecilia, Belen, and Caitlin. And we're all really excited to be putting on some amazing events for you all this year. We're extending invitations to any woman or underrepresented gender to join our community if you'd like to take part in building this community with us behind the scenes. Otherwise, we welcome any volunteers with setting up future events just like this. Our general meeting, or AGM, is next Monday. So if you'd like to join, please let us know in the chat or in our Slack, and we'll advertise it there as well, so you don't get to miss out and you can help join that decision-making process for our next year's committee, or this year's and next year's committee. Alrighty, so I now have the absolute pleasure of introducing our speaker for today, Dr. Shazia Rupal-Basundas, who is a former president and alum of the Our Ladies Melbourne community and committee. Shazia is a genomic epidemiologist specializing in pathogen genomic epidemiology and its applications in malaria surveillance. Her research integrates population genetics, bioinformatics, and epidemiology to design analytical and statistical methods that enhance our understanding of malaria transmission dynamics. She is particularly interested in leveraging genomic data to optimize malaria surveillance strategies in response to emerging challenges, such as increased localized outbreaks, drug resistance, and climate change. And through her work, she aims to bridge that gap between data generation, analysis, and real-world impact. She's also an avid R user and is passionate about open science, training and capacity building, and the development of accessible digital tools. She's also a consultant and instructor for Applied Epi, where she teaches applied epidemiologists and public health professionals to apply epidemiological methods using R. And she taught me pretty much everything I know. So today, Shazia will be presenting about PGEforge, a centralized open access repository for malaria genomic data analysis. So Shazia, please take it away and I'll stop sharing.
Thanks so much, Dion, and all of you for joining, and all the Art Ladies. It's so nice to see everyone. I'm glad time zones could permit me giving a talk today. And let's see if this shares and looks okay. Is that all right for everyone? Awesome. All right. Well, it's my pleasure to talk to you about some of the recent work, and I'm really speaking on behalf of a large group of people who have developed PGEforge, which I'll describe today. And yeah, I'll give my due thanks at the end to all of the people involved. So just to outline what I'm going to go through today, it's not a science talk because I know this is Our Ladies, but I will just give you little flavor on the why, on why we did this. So I'm going to give just a brief background on malaria for those who might not work in the space, on the surveillance landscape, and also on genomic epidemiology very briefly. So I don't expect it to be super technical, but I hope that it'll at least get everyone on board on why we need this. I'm also just going to give you like a flavor of what the gaps are in the current landscape and sort of why we developed PGEforge, who and how. So because this is an Our Ladies meetup, I'm going to really be giving you sort of a description of how we developed this via a hackathon that we organized, and also using Quarto actually as a tool that helped us develop all of this. So including these slides. So you'll see it sprinkled throughout. And then just a very quick where to now. And yeah, I look forward to discussions at the end, and hopefully I won't talk for too long. I could probably give a talk on each of these things for like an hour, so I'll try and keep it brief. All right, so to give you a bit of the background, malaria is a major public health priority. You can see here on the right, this is the most recent data on countries that are still endemic for malaria. So malaria, oh, can you see this? Is this changing?
No, it's not changing. Not on
Oh.
my end.
All right, one second. I think there's something weird's going on. So you guys haven't actually been seeing my outline. 1 second. Can you see that? Any change? Can you see that now?
Yeah, that looks great.
Well, I was talking to text, but I'll just skip over now. All right, you can see that now, right? Okay,
Yeah, that looks good.
Um,
Maybe full screen as well.
oh, yes, there we go.
Great.
How's that? Excellent. All right, it's been a while since I've presented Quarto slides, as you can tell. All right, so malaria is an infectious disease that's caused by Plasmodium parasites and is transmitted by a mosquito vector. So I was saying before in the map that you couldn't see, that you can now see, this is a map of all of the countries that are endemic for malaria, so anything with a color, basically, except for green. So green are those that were recently eliminated malaria. It's endemic in 83 countries, and half of the world's population is at risk. So as I said before, it is still a major public health priority. In 2023, there were 263 million cases around the world, and almost, or probably above, 600,000 deaths. And these are sort of lower limits on these estimates. Importantly and alarmingly, the global malaria strategy targets that we have set, or the WHO has set, are not on track. So there are some emerging threats to malaria control and elimination targets. There are many, but I'm just going to give you sort of two. One of them is that antimalarial resistance has emerged in certain areas of the world and is now emerging also in Africa, where the burden is highest. And then also we have sort of new mosquito vectors spreading that are also now, there's evidence that they're spreading sort of both drug and diagnostic resistant parasites, which obviously really threatens the gains that we have made so far. I couldn't ignore the current landscape either. And so I don't think I need to tell all of you that we have a really fragile global health funding landscape at the moment. And with these cuts to a lot of global health programs and cessation of many activities, cases and deaths are expected to increase this year. There's two recent modeling efforts, one by the Malaria Atlas project, which is based in Perth, and they estimated above 14.9 million cases. It's kind of misleading for me to say potential additional cases. What they did in their analysis was to say, if we continued, we could avert 14 or almost 15 million cases, but you can kind of infer the converse of that. Similarly, with colleagues at Imperial, we modeled the estimates through different methods, and our estimates came up with at least 85,000 additional cases just this year alone. So yeah, a dire situation, not the best way to start. Hopefully I'll end in a positive note. Just to get you up to speed on sort of what the technical strategy, when I talk about these targets for malaria, there is a strategic framework that was set out by the World Health Organization that includes 3 pillars: ensuring access to sort of diagnosis and treatment, accelerating efforts towards elimination. So if you remember that map, there are countries that have reached sort of that green status, but all of those other colors are countries that are trying to eliminate locally. And what I'm going to be talking about today a lot is one of the pillars, which is transforming surveillance into a key intervention. Now, I also don't think any of you, even if you don't work in malaria, has been able to ignore sort of pathogen genomic surveillance as a thing that has been gaining traction since COVID-19. A good thing that has come out of the pandemic is that this has led to a rapid scale up of sequencing infrastructure around the world, particularly in sub-Saharan Africa. And there's now newly established networks that strongly advocate for the use of pathogen genomics to help and aid public health decision-making. Now, a lot of these networks focus on pathogens with pandemic potential, such as this recently established network through the WHO. But there's also a big focus and a big recognition of the need to also focus on endemic diseases like malaria. So this is basically just to say that genomic surveillance is widely recognized as an important tool for our toolkit to control and eliminate malaria and also understand the situation. So what do we aspire to? That was like my 101 in like 3 minutes of malaria. What do we aspire to in sort of genomic epidemiology informing malaria surveillance? This is the ideal cycle. I'm going to take you through it. The idea is that it is a cycle, so it's not a linear process, but it's actually a cyclical process. So can you see my mouse? Okay, awesome. So ideally, we want to start with a public health question that is defined by both researchers and policymakers. A lot of this is very malaria specific, but I'll just give you one example. So, for example, should the frontline antimalarials be changed? And as I mentioned, there's sort of emerging drug resistance. And so are the antimalarials still working? Might be the big question that needs to be answered by a policymaker. We then have the process of data collection, which involves a lot of important aspects like what kinds of samples should we collect, how many samples, do we have a statistically robust study design? We then generate data, and of course here I'm focusing on sort of genomic epi, and so we're generating sequencing data basically, and then we're also probably collecting associated epidemiological and clinical data. And then we analyze this. Now, I'm not going to get into all of those analyses because I know this is not a genetics talk, but basically we need to analyze that data. We also then need to actually synthesize and interpret it in a way that is useful to those who are making policy decisions. And then we need to communicate them. So we actually need to have a conversation. So this is this kind of ideal framework that all of us and a lot of people online are working towards. And what we're trying to do and what we tried to do with PGEforge is try and fill some of the gaps that are existing in this cycle. So what is that current landscape in malaria genomic surveillance? I could also give an entire talk on this topic. So I'm just going to focus on kind of one big picture idea, which is there have been major advances in sequencing, as I mentioned, not just infrastructure, but also sequencing methods, bioinformatics pipelines, and also analytical tools. And here you can just see sort of ever since the Plasmodium falciparum or the malaria parasite genome was first published in 2002, there's been a spike in basically genomics papers, which has involved anything from understanding the basic biology to understanding transmission to developing new genotyping panels and bioinformatics tools. In parallel, there's also been an effort in conversation with sort of those policymakers at the other side of that cycle, how can genetic epidemiology be used and be applied for malaria elimination strategies? And this has defined sort of very key use cases, as they're called, or sort of applications of genetic epidemiological data where it can inform. And so if we continue with that example of drug resistance, sequencing data to actually sequence those drug resistance markers can give us a sense for how well our antimalarials would be working in an area, for example. There's also been a lot of recent efforts to provide more specific guidance. So I don't know if you can really see that, but this was a recent review on sort of how should we sample. So if you remember sort of that study design, if we have certain questions, how do we design statistically robust studies? What molecular markers should we use? So this is a whole other area of, if we're trying to look at drug resistance, then there will be certain parts of the parasites that we want to study and sequence to understand what's going on. And then also, how can we use molecular data? So this was also, I don't know if you can read that, but this was a recent review on like, how do we actually then integrate genetic data into modeling and more downstream analytics? All right, that was my then second 101 in 3 minutes. So what are the gaps? And this is kind of, I've tried to focus this talk a lot on the tools and sort of the R analytical part of this work. What are the gaps? We've actually done a pretty good job in the field of developing software tools to analyze data to support genomic surveillance. So there's lots of tools available, which is actually great. Now, there's little guidance on how to choose, use, or assemble those tools to translate genetic data into those interpretable and actionable results. So at that farther end of the cycle. There's also a lack of standardization of software tools, inputs, and outputs. And tools often still have limited usability by end users. So an aside here is that often many of the researchers who are developing these tools are not software developers. They're researchers. They're not trained in software development. They're not incentivized to actually maintain software. So that this process in sort of the field and in academia is sort of like, oh, I have this research question and I kind of made this method, and so I'm going to put it into an R package and publish the paper and never look at it again. And that's just kind of what happens, unfortunately. And there's many reasons why. And the main one is that as researchers, we're not necessarily incentivized to maintain software. We don't even really have a need to sort of have good documentation, for example, these kind of basic things that, you know, all of you in the R community would probably be excellent at. Another main issue is installation difficulties. So if you think about the end users of these tools that we're developing, a lot of them will be based in a malaria endemic country. They might not have the newest MacBook Pro, and you know, they might have some issues with dependencies or operating systems, and tools are not always tested on multiple operating systems. For example, a lot of these tools, because they're analyzing complex data, they often use sort of R CPP and maybe C++ interacting within an R package. And so you need a C compiler, for example. And so these are real issues that most of these tools have, unfortunately. And as I mentioned a bit, there's also non-standardized input formats. So all of these tools exist, but actually working with all of them is also very difficult. And these are very real pain points that people feel all the time. So our main aim is to fill this gap. And this has been through a community-led effort towards the foundation of an open analysis ecosystem for malaria genomic epidemiology. And as I kind of alluded to at the beginning, we did this through a hackathon approach, which we called Radish 23. And so anyone who likes setting dumb acronyms will appreciate this. But this hackathon was basically to try and make these tools more reproducible, accessible, improve documentation, and improve interoperability standards. the Plasmodium Genomic Epidemiology Network, which is a network of researchers across many institutions globally, convened 18 experts across 15 institutions, and we all came together in Baltimore in December 2023 now. Seems like a while ago. And to put it simply, our goal was really just to take this wide range of software tools that were available and make them easier and more straightforward to use as sort of a first step in trying to get to this open analysis ecosystem. So as I mentioned, this is a huge effort, and here are all the awesome hackers that were part of this event, including some people on the call who I can see. And the main goals for the hackathon are we had to do some pre-hackathon work. And so given all of these tools, no one has had done the task of like just landscaping them, like what is available, what is out there, what do they do, who's developed them. And so what we did was, actually, sorry, before that, we realized that if we wanted to do anything to improve these tools, we needed to kind of fix that input format problem. And so what we did was we compiled both simulated and empirical data sets of the types of data that would be sort of the common input to all of these tools. And we had to sort of map out what those input formats are and then come together to kind of generate these simulated and also just pull from the literature some empirical data sets. So this is now what I'll kind of term like the canonical data sets that are available for anyone to use if they just want to test things out as well. And then, as I was saying before, we also just had to sort of scope the landscape. And this is I purposely made this small because it was massive. And it's just to illustrate that we actually did quite a bit of homework before the hackathon. So everyone was tasked with like, literally it was like, put a row of everything you know that exists, and then we'll do kind of like a search. And this was obviously not like systematic review or anything. There could have been a way better way of doing this, but this was kind of our first pass at like, what is out there? What are the functionalities of these tools? And then this helped us identify tools that, for example, maybe are no longer maintained. So I'll give a quick example. Like one was like a .exe file that you downloaded and like ran locally. So stuff, as you die, and both like, wow, yep. There were other things that were called tools and were like an R script. And so there was several reasons that we kind of identified tools as like not priority. And then we also selected priority tools for focusing on during the hackathon and basically for resource development. So now during the hackathon, which was 4 days, we focused on developing material for each of those priority tools. So each hacker was assigned approximately 1 to 2 tools, depending on difficulty. Some were easier than others. As you can imagine, there was a wide range of types of tools available. More recent tools tended to be better. I think we have realized that this has been an issue in the last couple of years, but there's been tools developed, years before and stuff. And this material for every tool, we compiled sort of the information. So like having a centralized place with like all of the information, what's the paper, what's the GitHub repository, is it an R package, is it a Python script or, whatever. And then we also prepared installation aids. So as I mentioned, one of the biggest hurdles was actually just installing the software. So you can imagine you might have this like perfect R package that you've developed and no one's using it because they can't get it to run. Like that is just like the biggest nightmare ever, which was happening quite a lot, especially because of like C compilers and things like this. And so installation aids were developed for each tool, and I'll also get into how we tried to solve the installation problem in a bit. And then we focused on making these reproducible tutorials using the canonical data sets. So assuming I'm someone who wants to look at drug resistance and maybe I don't have data, but I want to test out how to use the tool, I can now use these canonical data sets. I can work through the tutorial. It's fully worked through from, how do I load my data? How do I look at it? What functions do I run? What do they mean, etc. And then we also had opportunities for sort of breakout discussions and mapping out analysis workflows, which I'll describe in a bit. And then we also had a manuscript writing team, which was basically the PIs and myself. And happily, I can say that this is now finally in preprint as of like a few days ago. So yay. I'm glad I had this talk because it pushed me to like actually meet that deadline. So just a break from me talking and just showing you like everyone sort of hacking and like it was very fun. We had a lot of candy, a lot of R on everyone's screens and some fun events too. In terms of the hackathon organization, I wanted to shout out Notion. I don't know if people love or use it, but we used we used this for organization, and like we had this kind of fun leaderboard of like, I mean, I should say we attempted to use it as best we could. Once you got into like the nitty-gritty, like we kind of forgot about it, but it was fun at the beginning. We kind of had a leaderboard of like who was doing what, like which tools are in progress, which ones are done, which ones are not priority, and etc. And like it was a good way of keeping track of where everyone was at and where things were. So now to probably what all of you are interested in, not nothing of what I just said, but probably this part is how did we use Quarto? I couldn't have a presentation without having some kind of Allison Horst illustrations because I just love them. But basically, I love this because it is exactly what we were able to do. We used, Quarto to develop these HTML tutorials. We also used it to develop our website. So this is kind of the GitHub repository that we set up before the hackathon, and I don't know how much interest there is in knowing how we did all of this. So if you have questions, you can ask me later. But basically, we had everything within one repository, so including the Quarto website, and then the tutorials folder where everyone sort of had every tool had its own folder, basically. And then the way that we did this was everyone who was working on a tool had a sort of specific tool branch, and then the website was built automatically every time we kind of updated the repository, which was really nice because we could sort of see the progress as we were going and sort of building up to like having these resources in real time. We developed very specific contribution guidelines, which I think is important when you're working with so many people. So we had sort of a main and developed branch. We tried to emulate sort of what real software developers would do, which none of us are. But we kind of had this main branch that was sacred and that would build, and that was sort of leading to the website, and the develop branch, which was like the kind of working branch. And as I mentioned, everyone kind of created their own branch for their tutorial and then pull requested into develop, then develop into main. So that's our understanding of like how real software developers do things. And I just wanted to just kind of highlight what one of those pull requests might look like. We kind of had this checklist for people to make sure that they were doing what they were supposed to be doing and describing their changes or linking it to an issue if relevant. And then this was kind of checked. I think I don't know, I think we had like almost 100 pull requests or something like in 4 days. It was like pretty insane, but I think it was a good way of kind of working simultaneously with so many people. So the Quarto website is what hosts all of these resources. And this is sort of the landing page. We have sort of the canonical data sets available here with description, the tool landscaping, software standards, which I'll describe in a second, because that was an important output of this hackathon, was that together we kind of came up with what the software standards should be to evaluate tools and what should we be striving for in our tools. And we drew inspiration from FAIR standards and from other communities that have these kind of standards. And then the tutorials and the analysis workflow. And I'm going to give you a quick tour. I kind of thought of doing a live demo, but I feel like I'll just show you static stuff. And if you're interested, we can look at it later. So I mentioned the installation being an issue. This was an exciting output. We now have sort of our own Plasmodium epi CRAN. So I don't know how much people are aware of like the R universe through our OpenSci, but it is awesome. And it basically it includes sort of already built packages that can be installed really easily. So you can see there's 21 packages just in sort of our network. And this is only those that are our packages. So unfortunately, we couldn't do kind of the same for things that weren't our packages. But this basically made that installation aid the easiest task in the hackathon, I think, because now people just need to use one line of code to install the package because they can install it pre-built from this CRAN repository. So this is just an example of one of the R packages that we've developed actually that needed a C compiler to compile the package locally. But now all of that has been circumvented by the R universe because one line of code, everything can be installed locally now. So kind of a simple output, but like massively solves a huge problem. In terms of the landscaping, so that first document you saw, which was like awful, this is now sort of the pretty website version of it. And it's quite nice because it lists all of the software tools, the authors, what kind of stage they're at. And this is obviously not interactive, but you can also see sort of the main applications of the tool, the website, the GitHub repository, etc. So to the software standards, and I'd love to hear thoughts on this if anyone has any, but we came up with these measurable criteria essentially to evaluate comprehensiveness of documentation, quality assurance, and maintenance of the tools. And we did this in terms of sort of an end user perspective, but then also a developer perspective. So these were the software standards that we came up for sort of required for evaluation of the tools. So in terms of user-facing, do installation instructions exist? Do usage instructions exist? Do tutorials exist? And if they exist, do they have like an explanation of inputs and outputs and a test data set in a worked example? And then, of course, are these test data sets available? So that was sort of from a user perspective. And then from a developer perspective, is it open source? Does it have software tests or unit tests? Do they report code coverage? And are there clear channels for software maintenance and issues? So this was a difficult one because sometimes people put their email and it's like, okay, I guess they're trying to be to have a channel for sort of discussion and stuff, but is that the best way? Debatable. And so we tried to be lenient in that. And then we also defined these sort of nice to have standards, but we didn't actually use them for evaluation, but they were just sort of suggestions. So like informative error handling or multiple languages for tutorials, using standard data input. So that's kind of not an expectation, but an aim because sort of we're trying to solve that problem as we go. And then in terms of developer facing, minimal dependencies if possible, modular codes, so sort of split into functions, and then well annotated code. So these are sort of not requirements, but they would be ideal to strive for. So we did evaluate the tools and kind of dire situation as well, but maybe good call to action for the community. And so we scored sort of 0 if the criteria was not fulfilled at all. 1 if it was kind of fulfilled, so like maybe tutorials exist, but maybe they're not that good or fully the criteria is fulfilled. So this is we don't want to name and shame. So this is just the kind of overall findings. In terms of user-facing, actually, we're doing quite well. User-oriented standards were generally met for most, except maybe the test data and results. And then in terms of developer-facing, all of them are open source, which is great. But then, clear channels, like I said, I think we were kind of lenient with kind of saying yes to that one. Software tests and sort of code coverage was less so. So I think there's still work to be done as a community to kind of improve our tools. And then the next functionality was sort of these reproducible tutorials. And again, I don't expect you to read this at all, but I'll just kind of take you through. The idea of this is that a user, as I mentioned, can kind of go in and follow along. So here we've got that wonderful one line of installation, any necessary libraries, some kind of description of what's happening, and then loading the data, etc. So this is really just the idea, is to have this kind of worked through tutorial from installation to like output visualization interpretation for people. And yeah, we use the standardized data set, and so that ensures that anyone can do this from anywhere, even if they don't have data. Okay, so we've improved tool usability. Now what? And one of the big aims of the hackathon was to kind of come together as a community and redefine those applications. So that initial paper that I showed was very kind of big picture, like, oh, this would be great and that would be great to use genetic data to inform this and that, but like, how do we actually do that? And so what we did was we defined, again, sort of use cases, but more in relation to sort of what are the analysis requirements. So like, what are the actual functionalities that we need to do to actually get the outputs that we want? And so again, don't expect you to read all of this, but really what we did was define these use cases here on the left. And I see some malaria people, so if you're interested, we can talk about that in the discussion. And then the analysis functionalities, which were also kind of not decided. I mean, they came about from the discussions. And then just kind of like a presence absence, like which functionalities do we need to actually apply or fulfill certain use cases. And some of them require, like this one, for example, we need a lot of functionalities. So the next step was, which tools actually perform these functionalities, and then how do we kind of map things together? So how will this be translated to support translation, sorry, that was redundant, of genomic data for decision making? And so let's take that sort of mapping out of what we need and actually map out the workflow. So this is just one example, but the idea was to kind of get to a point where we know what tools or what functionalities we need to chain together to actually answer the questions we're interested in answering. Now, all of these words are very malaria genetics-y specific, so I don't want you to focus on that, but basically the idea is just to show you kind of how we came up with these workflows, going from raw genetic data to sort of these estimated metrics that we want, and the functionalities that are needed to actually get there. So then this allowed us to kind of map which tools are available, where are the gaps. So some of the workflows are still not possible because the tools don't exist, for example. So in this case, the question, again, the kind of motivating example I've had throughout is, how prevalent is parasite drug resistance in my region, for example? This is the workflow that I need to do. We know what tools are available to do which functionalities. However, chaining tools together might still not be possible. So that was something that came out of this hackathon. We might need extra steps to link these paths together. For example, we might need bespoke code or new utility functions. But at least we know where we're going and we know where the gaps are to be filled. So before wrapping up, I just wanted to kind of reflect a little bit on this approach. And I think it's safe to say it takes a village to raise a platform like PGEforge. I would advocate for hackathons. I mean, I'm preaching to the choir here, but obviously this is a pitch to maybe the more academic community. But yeah, I think the use of hackathons, where sort of large groups of advanced coders can just kind of get together for a focused time and to get through these kind of tedious tasks. We weren't doing any like sophisticated code or anything. It was just kind of tedious, to be honest. But no one person could realistically do this. So just a back of the envelope calculation. And this is just the actual 4 days of the hackathon, not all of the pre-work before or anything after. It was more than 500 person hours. This would take one person like more than 3 months full time. And that's just impossible. There's no incentive, like I mentioned before. No one's really getting paid to do this. Like, this is still kind of volunteer work, volunteer time. And so this is kind of the only way to do something like this. Like, this landscaping, I mean, it would be, you might get like a student to do it, but like, it's just like not a fun project or something, and you don't want to be doing that. This is a bit of a word vomit, sorry, but I think the landscaping and sort of mapping everything out is like a huge output of this effort because it allows us to define what tools need to be chained together and how. And then PGEforge has now set the stage for a lot of future work towards this open analysis ecosystem built around good software. So hopefully improving existing software and developing better software, and also keeping best practices for data analysis in mind. So what's in store next? I can actually describe a very recent hackathon. So continuing on with the I guess root vegetable theme. We kind of had the next iteration or sort of the follow on to this, which was focused around workflows. And yeah, I mean, we just strung together words to be able to say wasabi basically. But basically it was this hackathon model was repeated in January 2025, and maybe you'll consider having my colleagues give a talk on wasabi. I won't give away the kind of end to this, but the focus was on now taking those workflows that we mapped out and actually building that end-to-end pipeline that we outlined previously. And we managed to build two of those workflows from end to end, which involved a lot more kind of new code development. So I think in terms of comparing the two hackathons, this was really like actually more code heavy and kind of complex coding as well, to kind of fill in those gaps to actually be able to chain things together, especially as I mentioned, with different inputs and outputs, like actually just translating from one tool to the other, like all of those utility functions was developed during this hackathon. We also have we now have the foundation to be able to benchmark tools towards best practices guidelines. So this is ongoing work, and we're planning another hackathon in the summer, not Australian winter, Western summer. And the plan is to benchmark the tools. So now we've mapped out the tools, we've mapped out the workflows, we know which tools do what, but how well do they do them? And how should they be used? And so this is, we're going to evaluate tool performance for the given functionalities, because in some instances, there's many tools that do the same functionality and they might be useful in certain scenarios and not in others. And then this is also ongoing work, which is kind of a longer term project, which is what happens after we've done all of this. Like, how do we visualize this? How do we get to that end, that kind of end part of the cycle? And how do we visualize this? And I mean, Di will call you when when we get to this stage. But yeah, I think that's a massive part of this that we're still sort of slowly working towards. And yeah, finally, I just want to repeat that it takes a village to raise PGEforge, and I just want to acknowledge and thank everyone who has been involved from across the world, and also to thank the funders, RIP, in some cases. So I think I just want to recognize that it's unusual for this kind of work, like methods development and tool development, and it's not really an academic exercise per se. And so it was really great to be able to have this work funded and supported, because it's like a major advance for the community. So yeah, thanks so much for listening, and I look forward to your questions.
Thank you so much, Shazia. That was an amazing and insightful talk. As a malaria researcher, I'm biased, obviously. So does anyone have any questions? Feel free to unmute yourself and ask a question.
Hi, can I go first? Is that okay?
I'm going to stop sharing so I can like see everyone.
Yes. Hi, Shaza. Nice to meet you. I'm very inspired by your talk because I'm kind of weird in the academia because I sit between the ecology space and I also have a side gig of being a bioinformatician in the human data. And I can see the stark difference. Like when I do my fish bioinformatics study, I'm suffering all the time because there's really not a lot of the things that you talk about, like no standardization, tools are like scattered everywhere. So I'm not sure if I would be able to pull off the things that you did. Like, as you have said, it would take me 3 months. But I would really, really love to start small. So my question is, how did you start with the landscaping?
Oh,
Like, where to start? Like, what was did you do a Google search or like, do you have like a criteria? Something like that.
yeah, that's a good question. I mean, I think for for our purposes, we leveraged the Plasmodium Epi network, which is kind of like it's kind of has solved the problem of kind of at least bringing together a lot of the major players in the community, you know, and so at least we kind of leverage that knowledge. A lot of the people in Plasmodium Epi and actually the people at the hackathon were like developers of the tools themselves. So we had like a requirement that people couldn't work on their own tool because obviously they're going to be like, oh, it's like done. Like it's super easy to use and install. And so I think that was kind of a solved problem maybe in that sense. But I mean, I would suggest, as I alluded to, I don't think we did like a very systematic way of doing this. Like it was just kind of like, oh, you know, we've got enough people to kind of cover most of the groups working in this space. Like, let's just all jot them down, which I don't think is kind of the best approach potentially. And so you might consider some kind of systematic or like scoping review maybe might be like there's actual, you know, methodologies to that that might be useful for you to kind of bring together that knowledge, yeah, via like a searching strategy.
Awesome. Have my work cut out for me. Probably I'll give up on that dream, but yeah, let's see what happens. Thank
Good
you.
luck.
I think I've got a similar follow-up question. Thanks, Shazia, for this such a comprehensive project. Oh my God. I'm curious whether do you see any if this set of platform is transferable to other type of areas. So for example, I'm currently in biostats and also a human epi kind of space. Do you think what this set of platform and work can be done in other space that helps like summarize the tools and improve all the tools altogether?
Oh, definitely. Yeah. I think it'll obviously the kind of the focus might be different, like depending on what the field is or you know the purpose of the tools, etc. But I think as a conceptual, like it's definitely applicable. And I should say, like we're not the first people, you know, we're not like the first people to do something like this. Like it exists across many fields. I would say like the kind of viral and sort of bacterial genomic epi communities are like way more advanced than the malaria community. If anything, we're like striving to like catch up to them. Like they have, I don't know how much you guys would have seen during COVID, but they have all these like beautiful, like centralized platforms of visualizing data and stuff. Like we're not there yet. And it's a more difficult problem in malaria, to be fair to malariologists. But yeah, like I think there's other communities doing it really well. And that reminds me, there's also a great preprint. I don't know if it's been published, but it was from the bacterial genomics, maybe. But you might be interested in that, Megan, as well, because they also had a hackathon and what they did was more on the bioinformatics side of things. So we focused kind of more on the downstream analysis, not so much on the bioinformatics pipelines, but they actually made all of the bioinformatic pipelines like interoperable. And so, yeah, I can send you that link because that was like a really cool, that might be more useful actually for what you need in bioinformatics. And I didn't even talk about this, but there's a whole other effort of like creating kind of a new file format in the field that's being led by some of my colleagues. And that's kind of in the similar spirit of like everything, whatever the input and output is, like go to this centralized and then like kind of go back out kind of thing. And so that, yeah, that might be useful as well in the bioinformatics space.
Yeah, because I know everyone here who uses R would know it's like a pet peeve of ours. Like each package would need like a different file format. I'm like, but that would be
Yeah.
useful.
It's like a real pain point that I think no one talks like, well, wants to talk about, but like it exists.
Yeah, half of the time you're just arranging tables for it to fit. But I would love that link. Thank you so much.
Thanks, Shazia.
Um, this is spectacular,
Shazia. Absolutely spectacular. So now I'm curious, how is your plan for maintaining it and for updating it on a regular basis?
Great question. I think luckily we have quite an active, like existing community of like initial sort of you know, first cohort of hackathon, if you will. We've presented this in sort of bigger forums in the field, and I think this is kind of the negative of any kind of community effort is that it will always be like a 20% of the people kind of leading it and taking it forward. So we have, it's like open source, anyone can contribute, but at the moment, it's still just kind of the core group still kind of keeping things going. Ideally, it'd be great if things could, if more people would kind of contribute and get involved. Like, for example, any new tools that come out, they wouldn't be in the landscaping, like someone will need to add them. I kind of feel like we probably have to tap on people's shoulders, like, would you consider adding your tool? And like, maybe someone else can do your tutorial or something like, but I think it still needs that like one or two or three people to kind of keep things moving along. In terms of funding though, we did receive, so some of the PIs on this grant received funding for this kind of ecosystem development. So there is at least money behind, which is why we can kind of keep hosting these hackathons and stuff. So I think there will be momentum, but yeah, that's always going to be a challenge, I think.
I have a follow-up on that. So, I mean, congratulations on getting that funding. That's incredible. But would it be, I guess, beneficial to you and the team to have software developers join that conversation as well? Is that something that you guys are looking into?
Yeah, so this funding actually will support a software developer. So a lot of those kind of gaps that were identified in terms of tools that might not yet exist, and also in terms of that sort of file format stuff that I didn't even get into today, that's all going to be paid time for someone who actually has the expertise. So yes, definitely. I don't think we should expect academics to like become software developers unless that's their interest. But then I think like finding some kind of middle ground is good. And especially, moving forward for any new tools, having that kind of experience, a person with that experience developing it is like the ideal.
Oh, sorry to jump in again. Are you planning to write like a paper that would describe the recipe for how you build PGEforge? And if you're not planning on it, please write it because I would love to have it.
Yeah, I mean, the preprint that we put out is kind of, I would say it describes the use cases and it describes the resources, but not so much maybe focus like what I focused my talk on today. Like I obviously
Like
went
a
into
framework.
more of
Yeah.
like the nitty-gritty, but I mean, I don't know, like what would a forum be where we could publish that? Like I would love to do that. I think it would be helpful, but it's kind of hard. Like maybe just a preprint, I don't know. Or maybe just even on the PGEforge website. But yeah, that's a good idea.
And bouncing off of Dai's question, because this is really an awesome idea. And I think if we can make it transferable, it would be like a very good tool to like, this is the recipe if you want to do it on your field. So step one, step two, step three. Maybe
Yeah.
a good funding opportunity as well.
Yeah, no, that's a great idea. I'll
Yeah,
have to have a bit of a research on like where
that's a
like
good point.
kind
Yeah.
of open source journal maybe, or like yeah, I'll definitely do some kind of search, or if anyone has any ideas. But yeah, I think that would be very useful. Or I guess it seems like it's useful, so I'd be happy to do it.
Sorry for giving you more work.
Alrighty, if there are any last burning questions, feel free to ask them now. Otherwise, we'll let our
I
speaker
should
off
have
the
put my
hook.
email on the slide, but yeah, you can circulate it if anyone has any questions or anything. I'm happy to chat further.
Amazing. Well, thank you so much, Shazia, again for this amazing talk and for spending your morning with us. And thank you everyone who came. We're going to be posting this recording on our YouTube channel, so that people who couldn't make it can come and see. So yeah, thank you very much, everyone.
Thank you so much. I'll be posting the slides as well, so you can share those.
Amazing. Thank you.
Thanks, everyone. Nice to see you all.